introns separated by 17 of these intron spacers. The ribosomes never see the introns. They will only see the exons. So this whole process that we have going from a copy of a gene that's done in the nucleus is complicated if you look at what happens to that transcript before the ribosomes get to it. Because what you'll do is initially copy a gene, just like we saw in the film. The gene will copy the entire sequence of DNA going from this promoter region all the way to the end, to a region we call the polyadenation signal. And that's copying everything, introns and exons both. It's just one long sequence of RNA bases. But then that gets edited before it ever leaves the nucleus. And those introns are removed, and some of the exons are rearranged. So you have a lot of editing that happens inside the nucleus before the ribosomes ever see this transcript. So you will have the transcript modified by putting a cap and a tail on it, so it has some stuff added on both ends, but then also all the introns are removed. Some of the exons may be removed, and others put together in different arrangements. The result is that one gene can actually code for a number of different proteins, because one gene can be used to make a variety of different transcripts that eventually get to the ribosomes. So just in a simple view, we get this. If we imagine the gene started with a promoter there, that's where the RNA polymerase will attach to, and it runs all the way down here. Well, the messenger RNA initially is going to go all the way from start to finish of the gene, and then some of the editing in the nucleus adds on the cap and tail. But then the introns get snipped out, the exons get put together, and this becomes the finished transcript that leaves the nucleus. And importantly, these exons are not necessarily all used or put together in the same arrangement. And any sort of rearrangement here leads to different instructions on how to build a protein. In more detail, we get this. We have a promoter, we have then a 5' end of the messenger RNA, or the DNA strand. Remember, we've got the 5' and 3' sugars. We always use those as a way of getting oriented to tell which end of a nucleic acid molecule is which. In this case, we're at the 5' end. Here we're at the 3' end. The entire gene is going from this 5' region, UTR's untranslated region. This is a short region just in front of the gene that isn't going to be used to make any sort of protein. But after that, we get into the first exon, intron, exon, intron, exon. Then at the 3' end of the gene, we have again an untranslated region that will not be used to actually make anything in a protein. And polyadenation signal, that is going to then lead to there being a long chain of A's put on. The tail that is put onto the end of the transcript includes a long sequence of A's, possibly hundreds of A nucleotides. They give basically a lifespan to this transcript once it's out in the cytoplasm. That once a messenger RNA is in the cytoplasm, it will start to be broken down by enzymes that are there. The messenger RNAs should not last forever, because the longer they're out there, the more proteins you're making, and you may not want an infinite number of a protein to be made. So you give these messenger RNAs a lifespan. One way to cut their lifespan down is to give it a shorter tail. Longer tail means they last longer in the cytoplasm, because they'll start to be digested at this end, and you have to chop through all the poly-A's before you actually start to break down the main part of the transcript. But the transcript is getting modified inside the nucleus, adding this cap on, adding on a tail, and then The tail, here we go with the polyase. Maybe this is 500 A nucleotides in a chain. You still have the introns and exons there, but now we have snipped out the introns and we've got a shorter transcript. This has been edited a lot now inside the nucleus to have this end where translation will begin, this end with a stop codon, followed by a long sequence of A's, again, to give this a regulated lifespan in the cytoplasm. So to remove the introns, we have these small ribonuclear proteins. They will combine along with proteins. They create these complexes that cause a small piece of the RNA molecule to bulge out. And what you do is a snip, snip right at those spots. You cut away the intron and splice the two exons together. This is called a spliceosome. So a whole complex of RNAs and protons, small nuclear ribonuclear protein particles, that was it. So RNAs and proteins together that are doing this snip-snip process to remove the introns, splice exons together, and the RNA gets to be shorter, with then this material drifting away and being broken back down. This is happening inside the nucleus. So what you do then is repeatedly do these little cuts, removing the introns over and over. This gets broken back down to individual RNA nucleotides, which can be reused in the nucleus. And we now have a shorter, more compact version of the gene. This is what actually gets to the ribosome. This is what leaves the nucleus. So starting from a long gene, we've made it shorter and we put on the cap and tail, and it's now a mature RNA transcript ready to leave the nucleus. Now you might wonder, why do we go to all that trouble? What's the point of doing all that? Well, this concept of alternative splicing came up when we were doing the work with the Human Genome Project. There were estimates that we, in our lives, we make about 100,000 different proteins as humans. If you look in different cells, different types of cells, at different stages of development, all the way from an embryo up to an old person, we make about 100,000 different versions of proteins. And the idea was that then we probably would have 100,000 genes as well, because studies with bacteria had produced this idea of the one gene, one protein hypothesis. Basically, it looked like one gene coded for one type of protein in bacteria. So the assumption was that that happened with us too. Assuming that we used 100,000 genes to make these 100,000 proteins, but actually we have about 20,000 genes, not 100,000. The numbers, the estimates of the actual numbers of genes have been shrinking and shrinking and shrinking as we've gone through the whole study of our genome, and it's much less than we once thought. So what we're doing is using one gene on average to make 5 different proteins on average. Some making more, some making less, but the typical thing would be one gene codes for 5 different proteins. You do that through alternative splicing, and that means that you're putting exons together in different arrangements and producing families of proteins. So one gene is able to produce slightly different transcripts that produce different versions of proteins that are related to each other, that have some of the same parts. But it's like you're going and taking some different parts and building several different machines out of them. They'll still have a lot of things in common, but they could do different things. And that's what happens with a gene that is being alternatively spliced. So you make different versions of proteins that all have some of the same things in common, but still have different functions. And one example here is this protein that shows up in our muscles, tropomyosin. So let's look at that. If you copy that gene just start to finish, making this free messenger RNA, this is copying everything, exons and introns too. That has 12 exons and 11 of these dark green introns, the spacers. And what happens is we have tropomyosin showing up in different muscle types. Skeletal muscle, that's the muscle that you use voluntarily, versus smooth muscle, which could be, say, lining your intestines. You don't have voluntary control of that. They both use different versions of tropomyosin, but they're coded by the same gene. What happens is that the skeletal muscle, tropomyosin, it's using these about 10 exons. It's not using number 2. It's not using number 12. It's using all the rest. And together, that gives you this compacted, mature mRNA once you put the cap and tail on there. And the ribosomes make the tropomyosin that works in skeletal muscle. But alternatively, there's a different version going in the smooth muscle. There you are using numbers 2 and 12, but then you're not using numbers 10 and 11, or number 3. So you end up with a lot of parts in common between these two proteins, but also some important differences. The result is that one gene tells you how to make two different proteins that are related but distinct from each other. And that's what happens in us. With that sort of thing taken into account, that's how we make 100,000 proteins with 20,000 genes. So that's part of what happens in us, and it's much more complex than what happens in bacteria, where they simply are using one gene to make one protein, and they don't use introns and exons. Virtually all bacterial DNA is coded. With us, very little of it is actually coded for proteins. Okay, let's pause for a couple of minutes, then we'll come back and do some more, look at the actual transcription process.